regular
But,
multiple
the
choice. Midterm is just going to be multiple choice. The unit 4B will be multiple choice and short answer as well. Next week, what I'm going to do is I'm going to give you some practice questions for unit 4B to get you back into the whole how do I take a multiple choice test again. And those will be reflective of the types of questions you may see on the exam. Nate?
The test next week, is there an essay?
There will be an essay. Or short answers, at least.
Yeah, that's my bad.
No worries. No worries. Yeah. Yes, I can.
It's too far
Christmas
away.
one?
Yeah. Thank you. Do you do like a cannon?
Do it for the recording too. Screaming goat. Gotta love it. All right, so yesterday we left off talking about how sometimes cells shouldn't go on and divide. And that could be because of something outside of the cell, their environment, external signals. So it could be because it's cold, there's no food, there's no water. All those cases would result in the cell going into G0, which is kind of a stasis. Hopefully it will come back out and receive the go-ahead and go ahead through synthesis and all that good kind of stuff. What I want to talk about today is what happens in cases where there is a problem. And this is where there's internal signals going wrong. An example of this might be if during metaphase of mitosis, spindle fibers are supposed to attach. And if they do not attach properly, when the sister chromatids get pulled apart, they may not be pulled apart properly. And so what can end up happening is if one spindle fiber doesn't attach, it could mean that both sisters get dragged to one cell rather than being split apart. If I was talking about us, 46 chromosomes, and 2 of those chromosomes went to one cell, how many would I have in that cell? 47. So I'd end up with one cell that would have 47, and I'd end up with another one that would have 45. The 45 doesn't typically happen. The 47 does sometimes happen, and that is Down syndrome. That's the most common one. In general, when this condition happens, it does not work very well for the cell, and the cell usually has to be broken apart. Yeah.
How could having 47 happen more than having 45?
45 results in almost always a death.
Like if
but 47 usually
if
survives.
Like 47 in one, wouldn't you have 45 in the other because they're splitting?
You would. I'm kind of jumping ahead of myself too because I'm really talking about not mitosis, but I'm talking about meiosis, which we haven't gotten to yet, with the sex cells. But you would end up with a case where you would have one extra chromosome, and that one extra chromosome, we don't know exactly why, but it leads to a problem with not understanding what's going on, which set of instructions to use because you've got that extra copy. But if a cell gets fertilized and you end up with 45 chromosomes, that usually just results in a spontaneous miscarriage. So it wouldn't actually happen. Does that make sense? Yeah. Okay. Yeah. So you're right. We would have both of these things. We would have a 47 and we would have a 45 produced. But in the case of a 45, this one usually just doesn't make it. It's just usually a miscarriage. Whereas the 47 goes on and there's some issues with the fetus, but you're still going to have a baby being born. So that can happen. There are specific external factors that can tell the cell, and these are not really environmental things. Well, they're sort of environmental, but they're still within the body. So now we're talking about mammals and animals, us. External factors that can influence the cell's decision to divide. One is a growth factor, or I shouldn't say a growth factor, is growth factors. So there's lots and lots of different growth factors, and those growth factors can be released by certain cells to cause other cells to divide, saying, okay, you need to grow right now. It's the time in puberty where we're hitting a growth spurt, and we need to divide and grow and divide and grow and divide and grow. So growth factors would be released. So there's something called human growth factor, for example, that gets released by cells to cause the cell division to occur. Platelet-derived growth factor is another one. And these are made by parts of your blood called platelets. Anybody know what platelets are used in? Platelets are produced when you cut yourself. So when there's some damage to your tissue, these guys will get produced, and one of their jobs is to help plug the hole. So as you cut through blood vessels, you're going to produce an opening that the blood can seep out or leak out and get into other parts of the body that we don't want it to get into. We want it to stay in that bloodstream. So platelets' job is to kind of stick to that opening, to that hole, and to help form a clot, so that now the blood is going to continue to flow through the veins and the arteries and such, and not through that hole. So if there is damage, if there's some type of tissue damage, platelet-derived growth factor is often produced to tell the cell, hey, we need to produce more of you. Can you grow and split and divide and grow and split and divide over and over again, so that we can get rid of this jagged hole that we have. Another external factor that can influence cell division would be density. So if there's a lot of cells around, that typically will cause the cell to second guess or to inhibit its growth, so that it's not going to continue to the point where it's got to divide and make new cells. It doesn't want to go through cell division if it's already crowded with cells. If you've got enough cells, you don't want to continue growing. I took out these next 4 slides. They're all pictures, so you can take those out. We don't really need to look at that. In addition to the 3 that I just mentioned for influencing cell growth, there's also some anchorage dependence, which basically means that cells tend to not divide if they don't have something to attach to. So they're saying they must attach to a substratum. That just means they have to attach to something. You don't want cells to just be floating around and not be anchored to anything. So there's some dependence upon, can they anchor or not? If they can't anchor, again, they're not going to, normally, they're not going to divide. Density dependent, this one here, density dependent and anchorage dependence will check the growth of cells so that there's an optimum level of density. So we don't have too many cells, you don't have too few. But sometimes this doesn't work. And so what we see in cells that have become cancerous is they do not adhere to this. They don't care if they can't attach to anything. And they don't care if there's already a lot of them. They just continue to grow and divide. Okay. So that's one of the problems with cells that are cancerous. And we're going to talk today about more of those situations where cancer develops. We can take this one out too because it's related to the last slides that we took out. And then we're going to go straight into how cancer develops. And it all has to do with that cell cycle. Okay. Cancer cells don't respond normally to the body's control mechanisms. Cancer cells do not need growth factors to grow and divide. So normally we need those growth factors like human growth hormone that will cause the cells to grow and divide and grow and divide. Cancer cells can make their own growth factor. Not only that, but they can actually make it seem like there's a growth factor there when there's not a growth factor there. Sometimes they just have abnormal cell cycles. Long story short, something's going wrong and they're not being kept in check like they should be with the cell cycle control systems that we have. Cells that acquire the ability to divide indefinitely are undergoing transformation. Cancer cells that are not eliminated by the immune system will form things called tumors, which is basically just a big ball of cells that don't really have a specific function. They're abnormal. All of them are descended from that first cell, and they don't really have normal function in the body. I think it is important to notice that the immune system actually there are immune system cells whose job it is to just look for these cancerous cells, to look for these cells that are not right, and to cause them to be destroyed. And not all of them, unfortunately, are found by the immune system, and that's when you start to get a mass of the cells. What happens when you have that mass of cells growing and growing and growing, they will start to press upon normal, healthy tissue. And that's when it becomes an issue. If a group of cells, if a tumor stays at its original location and it does not move, we call that benign. Okay, so if somebody says, I went to the doctor today, I had some cells removed for a biopsy, and they found out that they were cancerous cells, but they are benign. Okay, that's your best case scenario, because that means the tumor is localized and it's not going to spread to other parts of your body. So if that happens, that's your best case scenario. What you don't want to see, you can avoid, is malignant tumors. Malignant tumors are those that will spread to surrounding areas of the body. And they do that by what we call metastasis, or you might have heard that the cancer has metastasized before. And what that means is that the cancer, some of those cells from the tumor have broken off, and they've started to travel to other parts of the body. And they can do that through the bloodstream, or they could do that through the lymphatic tissue that we talked about earlier when we were talking about immune system. Malignancies are harder to treat because of the fact that now you're not just treating the source of the cancer, but you're also treating wherever it went, and you don't necessarily know where it went. It may be hiding somewhere, and you might not know it until it gets to the point where it's a big problem. Localized tumors can be treated with radiation, and basically you're just doing focused radiation on the tumor. That causes the DNA to mutate so badly that now the cells don't know how to even divide. So it basically stops the progression of the division, because now the cells can't function at all. Not only can they not stop growing and dividing, but now they're not going to divide at all. They just don't have any idea of how to do that.
Is it like it always makes it mutate to the?
Yeah, I mean it destroys it to the point where, I mean, maybe mutate might not be the best way of saying it. They affect it to the point where, the destruction of the nucleotide, so that there's not even a message that they can copy and do anything with. To treat metastatic cancer, so those that have spread, you usually use chemotherapy. So chemotherapy are drugs that target fast-growing cells, and they tend to be pretty invasive and pretty aggressive, meaning that they don't really recognize healthy tissue compared to cancerous tissue. They're just recognizing any cell that divides quickly. So people who go through chemo, one of the things they will happen oftentimes is they'll lose their hair. And that is because those hair cells are growing very quickly. And again, all the chemo is looking for is fast-growing cells, so they affect the hair. Same thing with the stomach. Sometimes you'll see nausea and really have a problem eating, because one of the other areas of the body that's very fast-growing is the lining of the stomach. That gets replaced every couple of days. So the body again goes, oh, yep, or the chemo goes, yep, that's fast-growing. So they're gonna destroy some of that too. Sometimes you get sores in the mouth as well. Again, fast-growing tissue is going to be eliminated. Doesn't matter if it's cancerous or not. So next picture is of a tumor in breast tissue. So there's a very small lump here to begin with. And this is why it's important that no matter where your tumor is or where, if you feel a lump, you should go to the doctor and get it figured out. Because what happens is if you let it go, it will grow and it'll get bigger and it'll get bigger. And if it is a malignant tumor, it can spread. So here we're seeing that it's getting bigger. It's getting bigger. And at some point, it's going to get close enough to either the bloodstream or to the lymphatic system. And when it does that, remember, these cancer cells don't have to anchor to anything. And so they can be produced and then they can just go into the bloodstream. If they're close enough, they'll just float into the bloodstream, diffuse into the bloodstream or the lymphatic tissue, and now they can move. Yeah.
This sounds kind of stupid, but if like cancer cells have no function, why is it that they're so?
That's a really great question that it took me a long time to figure that out too. It's like, well,
Yeah.
if they have no function, why is that a problem? It's not that how do I want to say this? Let me just give you an example. What they do is they will press upon tissue or damage or destroy tissue that's normal. So if you've got, if you have some of those cells that suddenly lodge in your brainstem that are responsible for respiration, for breathing, and they continue to divide and divide and divide, what they'll do is they'll press against and damage the healthy brain tissue so that it can no longer function, and then you can't breathe anymore. So that's the long story short of it. Yeah. It's this kind of insidious thing.
Yeah.
It kills us. It's our own body. It's our own cells, kind of, killing us.
Yeah.
So cancer, just defining it here, is unrestrained, uncontrolled growth of cells. The cell cycle doesn't stop. Okay, and so we've learned a lot about how the cell cycle impacts this, and we found that there are genes on these chromosomes that can be altered. And so when I say altered, I mean that they have been mutated. So mutated means that their DNA sequences have changed. So if it's an A, it's an adenine. Remember we talked about nucleotides back in the day? Oof. Sugar, phosphate, nitrogen base make up a nucleotide. Those nitrogen bases can be A, C, T, or G. Okay, pulling out the cobwebs from earlier this year. If that sequence is changed, then you're going to get a protein that's going to be different from what was originally supposed to be there. And that can cause issues. And so we're seeing in many cancers, there's really two types of genes that are affected and affect the cell cycle and therefore cause or let cancer take hold. And those are tumor suppressor genes and proto-oncogenes. So first, we're going to talk about tumor suppressor genes. So what do you suppose a tumor suppressor gene does? Suppresses tumors, yeah. So one that we're going to highlight here, and we'll later do a lab on this, gel electrophoresis lab, is P53. So P53 is a gene that codes for a protein that is important in looking at the DNA at G1. So before the cell, what's the cell going to do after it goes from G1? Where is it going to go? Into what? Synthesis. Yeah, it's going to go into synthesis. It's going to make more DNA. Now, the thing is, if that DNA is messed up at G1, the last thing you want to do is now make two copies of it, because now you're going to have two cells that are messed up. So what P53's job is to make sure that if the DNA is messed up, then that cell cycle is halted for that cell. And one of two things happens. It either initiates the repair of the DNA so it can go forward, or if it's just too bad, then the cell will be directed to commit apoptosis. Remember apoptosis? So basically cell suicide. Apoptosis. Okay. That then prevents more cells with that same altered DNA from going forward. Nick.
How does it how does like DNA get repaired?
There are enzymes whose job it is to check over that, and if they see it, they will actually take out the messed up DNA, cut it out, kind of like scissors, and replace it with the correct nucleotide. What happens just because a cell is damaged, it's not at that point cancerous, right? And it could still go forward, but over time, if enough of those mutations, those alterations add up, it could add up to a point where those cells now are going to be a problem. What happens in these types of cancer is the tumor suppressor genes themselves that produce this protein say, nope, stop. They get mutated. So now they don't work. Their proteins that are being produced don't have the correct shape. Because they don't have the correct shape, they can't work. So because they can't work, anything that's damaged goes ahead and goes forward. Mary. Ooh, that's a good question. I don't know that answer off the top of my head. I'm pretty sure it's out there. I don't think it's a lot. It might be like only 1 or 2%, but I mean, over the course of a lifetime, that's a lot of divisions. Yeah, I mean, I think we're pretty good at it. I think we're pretty good at checking our DNA right after synthesis and fixing it. But even with that said, that's still a lot of divisions. And they tend to add up over time, which is why people who are older tend to get cancer rather than people who are younger, because they just don't add up enough when you're younger. Good question. There we go. Let's try that. So this picture is just showing that in the bottom here, you can see that some of these cells are just messed up, and they're actually not cancerous yet, but then something happens, and now they don't abide by the cell cycle at all. Okay. Tumor suppressor genes are just one of many that control the cells and keep them from going forward when they shouldn't. Remember genetics? Do you remember when you had something that was homozygous dominant versus something that was heterozygous? Okay, good, because that's what we need to talk about. So in P53, both, it's in red here. I don't know why it's in red, but it's okay. In P53, you have to have both of those copies mutated. So if you're like this, you would be heterozygous, and you only have one bad copy. That small p I'm showing as a mutated P53 gene. But over the course of your lifetime, what can happen is that second copy could get mutated. So you could go from being heterozygous in a particular cell to now you could become homozygous recessive. Now you don't have a good P53 gene. And that can cause the cell cycle no longer be monitored. Yeah.
Why does cancer cells happen more when you get older? Like the risk.
So what happens is the older you are, the more cell divisions you've gone through. And so the more chances there are that something is snuck through and it just continues to get worse and worse and worse until it gets to the point where it's actually a cancer cell. So that's why. The first tumor suppressor gene that was identified was the retinoblastoma susceptibility gene. And let me see if I actually want to talk about this or not. It's a rare form of cancer in the eye. Yeah, okay, there's not much here. So if you inherit it, that means that you have one good copy left. During the hundreds of thousands of divisions that occur to produce the retina, any error that damages the good remaining copy could lead to a cancerous cell. Okay, so you're mutating the checker, and once you mutate that checker, now anything that was wrong in that cell is going to go forward. Questions?
Yeah, if you got like cancer in your eye, like radiation, would you like go blind?
My guess. So your retina's back here, so I'm guessing they could probably go on this side. But it's a good question because the retina is responsible for transducing light into something our body can, our brain can understand. So I don't know. Actually, it might. Yeah, Makani.
Kind of adding on, this might be a dumb question, but like, if you have like eye problems or if you have like astigmatism or you wear glasses, does that like make you more prone?
No,
Okay.
no, those are not. Anyway, we are good, Makani. We are totally good with our near blind astigmatism problems that I know I have anyway. Yeah, I feel you on that one. No, they are not related, fortunately. Okay, proto-oncogenes. I just think that sounds like some kind of sci-fi word. Proto-oncogenes are good, and their job is to basically turn on the cell cycle. You say, hey, it's time to grow. We cut ourselves, or we're in puberty and we need to, make some longer bones, for example. What's the study of cancer called? Oncology. Oncology. Because now, I'm sorry, that kind of looks like gynecology, weirdly enough. Sorry, it's oncology. Oncogenes. Notice the proto's not there anymore. Oncogenes cause cancer. Okay. These are genes that get mutated in a way that keeps the cell cycle moving forward. Rather than shutting off after you've grown and divided and grown and divided and now you're done, those oncogenes now don't shut off. Some of them code for signal transduction proteins. And these can be a little more devastating because you only need one copy of a proto-oncogene to be mutated, okay, in order for that uncontrolled division to take place. Anywhere, yeah. Not fun. The RAS proto-oncogene is found in about 30% of human cancers. The RAS gene produces a RAS protein. And the RAS protein is wait for it, it's a G protein. And that G protein is going to turn on a whole bunch of these things called protein kinases. So it's going to cause a phosphorylation cascade. So it's going to cause one after another to be turned on. Long story short, it's going to cause genes to be expressed that should be turned off. Let me see if I got that picture here. And the end result is you end up with a protein that causes stimulation of the cell cycle. So it continues to go through the cell cycle repeatedly when it should get turned off. So the response of the RAS proto-oncogene is it's a synthesis of a protein that stimulates the cell cycle to go forward. Many RAS oncogenes have a point mutation. A point mutation just means that there is literally one change in the DNA, like from an A to a T. And that causes the RAS protein to be hyper active, and that causes this excessive cell growth or cell division. So you can have tumor suppressor that no longer function and don't work to turn the cell cycle off, or you have the proto-oncogenes that get basically turned on and you can't turn them off. Those are the two different mechanisms, and they have the same outcome, but they just have a different mechanism for doing it. This is just kind of a breakdown of it, of the proto versus tumor suppressor genes. Growth factor receptors, RAS protein. So RAS protein is active it's mutated in 20 to 30% of cancers. I think kinase is associated with that RAS protein. And down here, the RB protein is mutated in 40% of all cancers. And This is the big one. P53 gene is mutated in 50% of all cancers. So these guys have a lot to do with why we get that abnormal cell growth and we get that cancer in the first place.
And can there be like multiple of those in one cancer? Like
Yeah, there can. And some people, like with the P53, some people were born with it; they're already heterozygous. So, and some people are born homozygous dominant. So, somebody homozygous dominant can still get cancer. They just have to have 2 hits to both of their copies. But it can happen. If somebody's heterozygous, you're already predisposed to it because you only need that 1 hit. Yeah. So they're now looking at targeting people's DNA specifically to try and treat particular tumors. So there's some personalized medicine going on. I haven't heard too much about that lately, so I don't know how that's going, how that research is going. But basically trying to see, okay, is it a messed up P53 gene? Is it an RB gene? What is it that's going on? And can we tailor the treatment to that particular mutation to deal with it most effectively? All right. That's all the notes.